So we are continuing our detailed look at A-level biology with now episode 4, where we are looking at the structure and function of organelles. So we've looked at the origin of the organelles. We have then looked at the protein synthetic organelles. We next looked at the mitochondria, and now we are looking at the chloroplast. In this video, what we are going to do is, as per our last episode, we will look at first the function of the chloroplast, what does it do, and then we will look at the structures of the chloroplast that allow it to do those things. It's going to be a little bit of a deep dive, so let us begin. First question, what is the purpose of the chloroplast being in the cell? So, if you think about the function of the cells, right? So what we have already discussed is that cell function, so cells work, and because cells work, tissues work, because tissues work, organs work, because organs work, organ systems work, and so on. But cell function needs cell activities. Okay, so there's lots of individual things happening in the cell for the cell to be able to work. Now, for all these individual things to happen, many of them need energy. Okay, so actually that arrow is going in the wrong direction. Many of the cell's activity need energy, and the energy comes from ATP hydrolysis. Okay, now ATP hydrolysis can only happen if there is something supplying ATP. Okay, so this energy is in the form of ATP, and the ATP production happens because of respiration. Okay, and specifically aerobic respiration is the thing that extracts the most amount of, or the kind of maximum amount of energy from glucose molecules, which is the respiratory fuels. And the structure in the cell that allows, at least in eukaryotic cells, that allows aerobic respiration to happen is the mitochondria. Okay, is the mitochondria. But the thing that mitochondria needs in order to carry out its function is oxygen, it is respiratory fuels. I'm going to try to tidy up my writing a little bit. Oxygen, respiratory fuels, and by that we mean things like glucose, we mean things like fatty acids. And this is where the chloroplast comes in, because millions of years ago, think about it, take your mind back to millions of years ago, when maybe the chloroplast had, the structures in the chloroplast had not evolved. Cells were swimming around the ocean, in inverted commas, floating around, okay? But they were going around the ocean, and they had to rely on finding these molecules wherever they were. And A, they had to find them, B, their concentration would have been really low around them in the first place. So it would have been really difficult to find things like this. Okay, or cells were relying on finding these things to survive, and if they didn't find those things, they would die, because they wouldn't have respiratory fuel to supply to the mitochondria, and then the mitochondria would not have a way to make ATP, and then there would be no ATP to carry out the cell's functions. Okay, so let's come back to this then. It would have been then, millions of years ago, it would have been some mutation or something in the cells that would have occurred to start to develop these structures that would allow these cells to start making their own respiratory fuels. So if a cell mutated, developed some structures by accident that allowed the production internally in the cell of things like glucose, things like fatty acids, then cells would no longer be reliant on finding these things from the environment. And if that was the case, those cells that could build their own glucose, fatty acids, those cells would have a survival advantage on their competition. And they would have a better chance of survival and pretty soon you'd see many cells with similar structures. So that's where the chloroplast fits in functionally to the story of the functioning cell, because these molecules, such as glucose and fatty acids, they are provided by the function of chloroplasts. Okay, so autotrophic organisms such as plants, for example, they have chloroplasts, and so they can build glucose and fatty acids for themselves. Heterotrophic organisms, animals, etc., they need to consume autotrophic organisms, indirectly or directly, but they need to consume them in order to get these molecules. Okay, so you could argue, well, why don't animals have these photosynthetic organelles? We don't have them because we consume we consume autotrophic organisms. Let's not go there. Okay, so let's just look at that diagrammatically. Okay, we're still rolling. Okay, so diagrammatically, what we're saying is that the job of the mitochondria is to produce for us, or to produce for the cell, ATP. As a byproduct, it also produces wastes such as CO2 and water. But in order for it to do that, it needs an input of respiratory fuel in the form of glucose or in the form of fatty acids. Okay, that's wonderful. But where do these come from? Okay, now the only way you can get them in a reliable and kind of relatively constant way is by having chloroplasts. Okay, so chloroplasts are the machine that produces for us these molecules. Now, in order for the chloroplast to work, it does need an input of inorganic molecules. It does need CO2, so it gets that from the environment, but it's inorganic. It does need water. Okay, but give it that and give it some form of energy, okay, and it can take all those things, put it through the metabolic processes happening inside here, or put it through the processes happening inside the chloroplast, and it will churn out glucose and it will churn out fatty acids. Okay, and this is really important, because not only is the chloroplast responsible for making respiratory substrates, but if you think about it, all the molecules in the plant can be produced by what the chloroplast end product is. So if you take sugars from the product as a product of photosynthesis, those sugars can be used to synthesize nucleic acids, DNA, RNA, okay, at least the monomers of that. It can be used to synthesize the amino acids, which can then be the monomers to synthesize proteins. Obviously it's a sugar, so we can also make carbohydrates from that, and we can also synthesize lipids. So the chloroplast is essentially the biological molecule factory. Okay, so it produces them from scratch in autotrophs, and for all the other organisms that can't make them from scratch, we just consume the autotrophs and ingest those or consume those biological molecules, and we get those biological molecules that way. So the role of the chloroplast is very important. Now let's begin to start looking at the structure-function relationship. So that was basically the introduction. But now what we're going to do is start looking in detail at the structure and function of the chloroplast, and we're going to do that in 3 stages. First is we're just going to quickly do an overview of the structure, so that when we are talking about the function of the chloroplast, we know the locations roughly of everything. Once we've done an overview of the structure, we'll look at specifically how photosynthesis works in this chloroplast as an overview. And once we have discussed how photosynthesis works, within the chloroplast, then we will look at the detailed structure of the chloroplast that allows all those individual stages of the photosynthetic process to function. Okay, so let's get to it. I'm leaving the mitochondria here because there's a lot of parallel structures within this, bugging me. There's a lot of parallel structures between the chloroplast and the mitochondria that, oh, that's also bugging me. There's a lot of parallel structures between the chloroplast and the mitochondria that it might help to just leave there and show the parallels. Okay, so let's begin. Overview of the structure of the chloroplasts. Just like mitochondria, they're an enveloped organelle, so both of the mitochondria and the chloroplast have an outer membrane. So if you want to be specific about it, you can say outer membrane of the chloroplast or outer membrane of the mitochondria. Okay, but they don't have unique names from that perspective. Okay, next, they also both have this space in between those two membranes called the intermembrane space. Okay, next, this is going to get messy, isn't it? Next, they both also have the inner membrane. Okay, so they both have this inner membrane. Okay, and hopefully you remember our discussions about, you know, the fact that the mitochondrial outer membrane and inner membrane have very distinct properties and functions. Okay, some related. So we've got this inner membrane. Then within the inner membrane we have the fluidic region. Now in the chloroplast, the fluidic region is called the stroma, whereas in a mitochondria that fluidic region is called the matrix. Okay, next, and this is where well, actually we could say that there's difference, but they are also related. Like in the mitochondria, the inner membrane is folded to form these structures called crista. Now, whereas the inner membrane in these regions is mainly involved in transport of things in and out of the mitochondria, the crista regions are the ones that are mainly involved in the the aerobic stage of respiration, the oxidative phosphorylation. Bear with me here, guys. Okay, now this is the thing, this is interesting to me, that in the chloroplast there's this extra membranous compartment further in, which is called the thylakoids. Okay, and so these are thylakoid membranes. But it's the thylakoids that are mainly, their purpose is the light-dependent stage of photosynthesis. Light-dependent stage of photosynthesis. Now, why that's particularly interesting to me is because the membrane of the thylakoids shares very similar components to the membrane of the crista. So, I'm just kind of theorizing here, but I kind of tend to think of the thylakoids as maybe bits that continued their journey of independence and just became fully independent. And, so instead of still being attached to this membrane, they kind of fully detached themselves from that inner membrane and became independent membranous structures within that fluidic compartment. Okay, further specializing themselves from or differentiating themselves from the rest of the membrane, which had a different function anyway. But anyway, that's just how I see it, and that's how I make sense of it. Just put those crista back where they came. Okay. Okay, so we have the thylakoids. Right now, the thylakoid membrane has a special organization. So all of this membrane, all of these membranes are called thylakoid membranes, but collectively, so this is a flattened sac of a thylakoid stacked upon another flattened membranous stack of a thylakoid. So collectively, this stack and this stack, they are called grana. Okay, and maybe one stack is called a granum. Okay, so these stacks of thylakoid membranes are called grana. Now, other interesting or points of note are the circular DNA. So the circular DNA is also shared between, or not shared, but they have in common the mitochondria and the chloroplast. Because remember, at some stage we've discussed already that at some stage, these structures have evolved from free living independent cells, and so they have their own genomes. Okay, so we have our circular DNA. Okay, other kind of important structures are these. Okay, so these are lipid droplets. Okay, and again, when we go into the structure and function of the chloroplast, we will explain the presence of all of these things. Okay, and finally, we have here the large starch grain. Okay, so those are the main structures of the chloroplast. And now what we'll do is, now that we know what's what, we look at the function of the chloroplast in terms of photosynthesis, and then we'll start looking at how, how these structures are adapted to allow the chloroplast to perform photosynthesis. All right, guys, so now we are going to talk about the function of the chloroplast, which is photosynthesis. So remember that photosynthesis needs carbon dioxide, it uses water, and with an input of energy, it produces glucose and oxygen. Okay, so this is what we are looking for the process to do overall. But obviously it's a metabolic process and it's going to do this in many stages. Okay, now there's two main stages in photosynthesis. The first stage is the light dependent stage. The second stage is the light independent stage, or sometimes called the dark stages of photosynthesis. Now the light dependent stage uses the energy in light to light energy is used or captured, and that energy is used to essentially produce ATP, okay, and produce reduced coenzyme NADPH. So essentially we've produced ATP and we have collected hydrogen. Okay, now in the light independent stage, that is the stage in which carbon dioxide is converted into sugars or organic molecules. So we go from inorganic to organic. Okay, but in order to do this, we need to provide ATP. So ATP needs to be provided in this stage, and hydrogen needs to be provided. So the ATP that was produced and the hydrogen that was collected is used up in order to convert carbon dioxide into organic sugars, and then as a product of that, then we can start making carbohydrates, we can start making lipids, we can start making nucleic acids. But overall, these two stages is, or combined, are, or is what results in photosynthesis. Now let's look at where and how these things happen. So the light-dependent stage happens in the thylakoid membranes. So I've redrawn the diagram here, and those stacks, those grana, the thylakoid membranes, I've redrawn here. Okay, and these are protein complexes within the membrane. This is the stroma. This is the inner chloroplast membrane, that's the intermembrane space, and that's the outer chloroplast membrane, and then outside of that we have the cytoplasm. So the thylakoid membrane is where the photosynthesis essentially begins. Okay, and first thing that happens is, well, let's look at the components first. So we have these photosystems that contain the chlorophyll. So photosystem 2, photosystem 1. Now the purpose of the thylakoid membrane is to capture the light. So these photosystems, what they do is, what happens is that light energy is absorbed by the photosystem, and what it does is it takes water. Remember, we use water. So the photosystem in the thylakoid membrane uses the energy of light to split water into protons and electrons. So I'm not going to worry too much about the numbers here, but we have an electron and a proton. Now what happens is that as a result of this, the electron moves into the photosystem and into these electron carriers. So again, this is similar to what we had in respiration. We have electron transport chain complexes that use the energy of using the energy of electron transfer to carry out some function. So we have the energy of light being used to split water, producing for us an electron and a proton. The electron becomes excited and starts moving through the electron transport chain. Okay, now, as in respiration, the electrons lose a little bit of energy every time they go through one of these electron carriers. Okay, now the reason why that is because the electron carriers are using the energy of the electrons to pump protons. Okay, so every time the electron, when the electron was transferred here and then moved there, proton pumped in. Proton pumped in, and then from this electron carrier to the next one, proton pumped in. So what we're doing is using the energy of light essentially to build a proton gradient. All right, so thylakoid membrane, key stage or key thing that is done is use the energy of light to build a proton gradient. So we've got much fewer protons in the stroma now, and many protons, relatively many more protons in the thylakoid space. Okay, and we use the energy of light to do that, because without the energy of light we wouldn't have had any excited electrons moving through the electron carriers. They wouldn't have therefore had any energy to pump protons from the stroma into the thylakoid space. So that's the first thing that happens. Okay, next, our low energy electron now is again excited. Okay, using energy of light. This time it comes to photosystem 1, the electron is again excited, but this time it's excited so that this electron can be combined with a proton from the stroma, further reducing the stroma proton concentration, and the electron and the proton combine and get added onto the electron carrier NADP to form NADPH. So we recombined the proton and the excited electron to form hydrogen, and the hydrogen got added to NADP to give us NADPH. Okay, so we're using the energy of light to build a proton gradient. We are using the energy of light to transfer hydrogen from water to NADP. Very important. So this hydrogen, essentially we can say that it came from the water, okay? Because remember at the beginning we produced a proton and the electron from water. Okay, another key thing that happens in the light dependent stage is we have to produce ATP, as we discussed. So as in respiration, building the proton gradient up means we have a now much bigger proton concentration in the thylakoid space compared to the stroma. So the protons cannot diffuse across this membrane anywhere else apart from through the ATP synthase complex, also in the thylakoid membrane. So the protons diffuse out along or down the electrochemical gradient through the ATP synthase, and as this is a passive process, energy is released. The energy released is captured by the ATP synthase and used to phosphorylate ADP to ATP. Okay, so that's another thing that happens in the light dependent stage, is that the proton gradient is used to produce ATP through ATP synthase. So those are the key things happening in the light dependent stage, okay? And as we said, it's this NADPH and it is this ATP which are then used in the light independent stage of photosynthesis to produce glucose for us. So let's see how that's done. Well, now we come to the light independent stage, the second stage. This happens in the stroma. Okay, so again, like respiration, there was one part that happened in the membrane, okay, and the second part or another part that happened in the fluidic part, the metabolic pathway. Okay, so one part dependent on electron transport, another part dependent on a metabolic pathway. And the same thing is true here. This metabolic pathway happens in the analogous part of the chloroplast, which is the stroma. So the light independent stage happens in the stroma. Again, it is a metabolic pathway. So mainly it's carried out by enzymes floating around in that fluidic portion. Okay, so, but what is it that it does? Okay, now again, it's a cyclical. This cycle is called the Calvin cycle or the Calvin-Benson cycle. Okay, so you've got a number of enzymes which carry out reactions, converting substrate to product, and the product of one enzyme is the substrate of the next one, which is, and so on. All right, but what it does is it takes CO2 from the atmosphere. So CO2 coming in, dissolving in the fluids of the plant, then making its way across membranes into the chloroplast, and it gets combined to molecules, pre-existing molecules, okay, and a number of reactions happen here. I don't want to get too much into the detail of this process because it doesn't really serve our purpose at this point, But what comes out, the product of this metabolic pathways, are 3 carbon sugars. 3 carbon sugars, and these 3 carbon sugars get converted to a number of things. So we might make glucose out of those 3 carbon sugars, and the glucose get converted to starch. The 3 carbon sugars may be used to make fatty acids, therefore making lipids or enabling the cells to make lipids. The 3 carbon sugars might be used to make amino acids and therefore proteins. The 3 carbon sugars might be used to make nucleotides and therefore make for the cell nucleic acids. So that's the point there, but key thing being that what drives this metabolic pathway and what allows each of the stages to happen is the constant input of ATP produced in the light dependent stage. So ATP is going in, and what keeps this cycle going is the provision of hydrogens from NADPH. These are the key things that we need to understand at this time. Okay, that this Calvin-Benson cycle that produces the sugars, that converts inorganic carbon into organic carbon and provides for the cell respiratory fuel, provides the cell the building blocks for its biological molecules, is only possible because of the supply of ATP and NADPH from the light dependent stage. The light dependent stage only possible with a constant supply of water and light. Okay? Yeah. Oh, and just as a by the by, this photolysis of water, the splitting of water into electrons and protons, at the same time produces that waste product essentially of this process, but quite important, generally speaking, oxygen. Okay, so that's the overview of photosynthesis. Okay guys, so let's just quickly recap that then. The light independent stage happens in the stroma. It's a metabolic pathway. Okay, and what it does is it takes in carbon dioxide, atmospheric carbon dioxide, and converts it into 3 carbon sugar. Okay, but in order to do that, it requires ATP. So essentially what that means is that the enzymes that carry out that process, those enzymes won't work without ATP, and they won't work without the hydrogen provided by NADPH. Okay, and there we have it. Now let us look at the structures, the adaptations of the chloroplast that allow those processes to occur. About 30 minutes later, Dr. Bhavsar gets to the point, the structure of the chloroplast and how the structural elements allow, or what role the structural elements have in the function of the chloroplast. Essentially, all the proteins that are present in the chloroplast, they have 2 sources. The first is obviously the circular DNA that is in the stroma. Okay, so I guess that counts as a stroma function. Okay, so the stroma has the circular DNA, and because of that, the stroma contains genes needed to make some of the chloroplast proteins. Okay, but I guess another additional to that is that in order to get these genes that are on this circular DNA produced into proteins, in order for that to happen, you need other proteins. So you need RNA polymerase to transcribe those genes, copy them into messenger RNA. Really? Okay, there does seem to be some kind of aircraft flying around, ladies and gents, so do bear with that if you can. Right, so the proteins in the chloroplast have two essential sources. So you need RNA polymerase to produce the mRNA, but you also in the stroma then would need some ribosomes in order to use that mRNA to make some of these proteins. Okay, so you need ribosomes as well. So these are all structural features of the chloroplast that allow it to have the important proteins that it has in order to carry out its function. So these enzymes, as part of the Calvin-Benson cycle, they are possibly, some of them are there because they were produced from genes on the circular DNA. Next, we have also discussed that as a consequence of this symbiotic relationship between this structure and the rest of the cell, part of that relationship is that many of the proteins required for this chloroplast to function get imported from the outside, or imported from the cytoplasm, because some of these proteins are, shall we say, immigrant in nature, and so they are effectively synthesized from genes in the nucleus, okay, perhaps produced in the cytoplasm as polypeptides, and from the cytoplasm then those proteins are imported. So those polypeptides are imported through the outer membrane, through the inner membrane, and then perhaps into the stroma, or perhaps through further through the thylakoid membrane and then into the thylakoids. Okay, so these proteins have to be imported, or some proteins, in order for all the chloroplast proteins to be here, some of them need to be imported from the cytoplasm, and that is where the outer membrane and inner membrane, and potentially even the thylakoid membrane, that's where they come into it. So in order to do this, you need specialized transport protein complexes. Okay, so these protein complexes, so that each of these blue squares could represent many proteins come together to perform this function. But if we did have a polypeptide that was outside, it would be the job of this protein complex to pull that polypeptide first through the outer membrane of the chloroplast, then through the inner membrane of the chloroplast, and then if it's a protein that ends up working in the stroma, then in the stroma it would fold up into its tertiary structure and take part in the metabolic reactions happening in the stroma, or this polypeptide might further then be required to enter the thylakoid or the thylakoid membrane, in which case proteins on the thylakoid membrane would be responsible to import that polypeptide into the thylakoid membrane, where it might function potentially as an electron carrier or a photosystem or ATP synthase. Okay, so that's essentially how the chloroplast gets its protein content, and the proteins determine the structure and the function of the rest of the chloroplast. So the outer chloroplast membrane and inner chloroplast membrane, their job is essentially to make sure that the appropriate transport occurs across the boundary that separates the chloroplast from the rest of the cell. So if we want to simplify it, we can say that it contains transport proteins. Okay, primarily what we discussed is that these transport proteins enable polypeptides, polypeptide import. Okay, so pulling the needed proteins from the cytoplasm into the chloroplast, polypeptide import. But what we must also remember is that the chloroplast is a biological molecule factory for the whole cell. So the things that it produces as a result of photosynthesis, it's not just going to indefinitely store them within the chloroplast. So likely these transport proteins also enable the appropriate transport of photosynthetic products, right? So glucose, fatty acids, it likely has transport proteins to enable the export of those things into the cell because obviously any lipids that are produced as a result of this need to somehow get to the endoplasmic reticulum so that those lipids can be used, so that those lipids can be used for the synthesis of membrane, thereby providing membrane for the rest of the cell. So we can't forget that. So we've got transport of photosynthetic products such as glucose, such as fatty acids / lipids. All right, so that's essentially the two membranes right there. Their key function is transport. Okay, now what we will do is look at the thylakoid membrane. Essentially what that means is, what are the structural adaptations that allow the light-dependent stage to occur? In terms of the light-dependent stage, there's 4 main structural adaptations that I think are important to mention. First is the presence of the photosystems. So in terms of what we discussed, the photosystem is the light harvesting bit. It's the part of the thylakoid membrane that allows the thylakoid to make use of light energy, to capture it and use it. 2, we next have the electron carriers. So the electron carriers are what build or use that energy collected by the photosystems in order to build that proton gradient. Okay. 3, we have the ATP synthase, which is essentially what makes use of the proton gradient. And 4 is really to look at the organization of the whole thylakoid membrane structure in terms of surface area. Okay, so then we just kind of take a zoomed out view. With regards to the photosystems, they are embedded in the membrane. So if that's the membrane, the photosystems are essentially protein structures that are embedded into the membrane of the thylakoid. But within that protein structure, you have specialized regions. One region, so first structural point is that it is a protein. Second structural point is that it contains this area called a reaction center. The reaction center contains the pigment known as chlorophyll, and it's the chlorophyll that is the light capturing molecule. Okay, so that's a structural feature of this photosystem, is that the reaction center contains chlorophyll, right? And it's the chlorophyll that acts as the light harvesting part of this reaction center and causes the electrons to become excited. As well as the reaction center though, in order to kind of maximize how much energy, because you've got energy kind of, lots of light energy is passing through this photosystem. In order to make use of more of it, and in order to make use of potentially other wavelengths than the chlorophyll alone can absorb, you have these other areas of the photosystem that have other pigments which can capture some of this other energy that or other wavelengths of light that's passing through the photosystem, but instead of transferring that energy to electrons like the reaction center does, these areas absorb the energy and pass the energy on to the reaction center. These other areas are called antennae. antenna complexes. Okay, now they also contain pigments so that they can absorb light, but instead of themselves passing that energy onto electrons and exciting them, they pass that energy onto the reaction center so that the reaction center can do that part of the process. Okay, so in terms of the photosystems, that's really all I want to talk about. The photosystems have the job of capturing light and transferring it to electrons and sending those or allowing those electrons to be passed on to the electron carriers. Okay, so the presence of the photosynthetic pigments is important. When we discuss photosynthesis in a bit more detail, like actually the process of photosynthesis, at that time we'll go into more detail about chlorophyll, electron carriers in a bit more detail. But for now, we're just focusing on as much detail as we need to understand the structure of the chloroplast. So that is the photosystem. Now we will look at the electron carriers. So the electron carriers are a series, again, they are primarily protein structures, but they are proteins that have transition. So let's just make a note of these key points. They are proteins primarily, okay, but they do have cofactors, okay, so they do have transition metal cofactors, right? Similar to, in fact, very similar to hemoglobin, for example. So hemoglobin has the heme group, and at the center of that heme group, you've got that ion. So similarly, these proteins you can think of in a similar way to hemoglobin. They have got these prosthetic groups, and they've got these cofactor ions that gives them the ability to carry electrons and transfer them on, okay, and harness the energy that's released when they leave. Okay, so what happens is that, so because of that protein nature, so let's keep the idea that they're proteins and the idea that they can carry electrons. So this part of this transition metal part of the structure is related to its ability of doing electron transfer. So that transition metal ion is related to electron transfer. So the electron comes in at high energy, excited by the photosystem, but in moving through, it loses energy, and all the electron carriers do this. Okay, so as the electron is transferred to the next electron carrier, it's leaving with a lower energy. And it's the protein part of the electron carrier that is able to do something with that energy that it has captured. And in the case of photosynthesis, pumping a proton from the stroma into the thylakoid space, as we discussed before. Okay, so the way I make sense of this is I say that the transition metal part of it allows it to act as an electron carrier, and the protein part of it allows it to act as a pump, moving H+ ions from the stroma into the thylakoid space, independent of the concentration gradient. That's how I make sense of this part of it. Okay, and all the electron carriers do that, allowing that proton gradient to be established across the thylakoid membrane using the energy provided by the photosystem. Okay, so essentially that's the electron carriers. We've looked at the photosystem harnessing light. We've looked at the electron transport chain using that energy of light to build the proton gradient. Now we look at the ATP synthase that's going to make use of the proton gradient. So let's look at its structure, very quickly. So we have that thylakoid membrane. We have the stroma here and the thylakoid space there. Now the thylakoid space, remember, has lots of protons, and the stroma has very few protons. And this is the gradient that's been built up by the electron carriers. Now in the membrane of the thylakoid is one part of ATP synthase that allows protons to move through down their concentration gradient into the stroma. Now, as they do this, they cause this structure to rotate. And as this structure rotates, it's connected to the outside part of it. I've kind of drawn it roughly like a mushroom, okay? But as the inner part, the bit that's embedded in the membrane, as it rotates due to the diffusion down the electrochemical gradient of protons, it causes the membrane embedded part to rotate. As the membrane embedded part rotates, it causes the outer part, or the bit that's in the stroma, to change shape, okay? And as it changes shape, those shape changes are utilized to carry out the catalytic reaction of adding a phosphate group to ADP. So ADP plus a phosphate group is added on to produce for us ATP. Now, those shape changes were only possible because this bit's rotating. And this bit's rotating only because there's protons moving down through it. And the only reason those protons are moving down is because there's a gigantic gradient, electrochemical gradient of protons. Okay, so that's kind of a brief look at the structure and function of the ATP synthase. But put those all together and you have light energy being used to make ATP. Okay, and so that is the ATP synthase complex. So we're on point 3, and essentially the fourth point was the surface area, right? So let's get rid of this. The fourth point is that the surface area. So clearly this is a process that is using light energy to produce ATP for us, but the biological concept is that the more of this stuff that we have, the more ATP, the more light we can use, and the more ATP we can produce. And so the more of this stuff that we can pack into this chloroplast structure, the more light we can use and the more ATP we can produce. And so we have the organization of these thylakoids into flattened membrane structures. Okay, and because of that, it increases the surface area. And as we've increased the surface area, we can have more photosynthetic components in the same amount of space, and because of that, the photosynthetic process can be more effective, efficient, or productive. And there we have the main adaptations of that allow the light dependent stage to happen. Okay, let's look at this actually. Let's do this. Let's do this like light independent stage and post light independent stage. So the light independent stage and the stuff that happens afterwards. Okay, so what structures are allowing that to happen? Well, the first one is that we have the enzymes. So all the enzymes, or the enzymes needed, all the enzymes needed for the Calvin-Benson cycle to occur are located in the stroma. Okay, so that's an adaptation. 2, you have enzymes here that are converting, so enzymes that are converting the 3-carbon photosynthetic product. So the main product of the photosynthesis process is that 3-carbon sugar. You've got enzymes converting the 3-carbon sugar into things like nucleic acids, into glucose, into fatty acids, into amino acids. All those enzymes are present also in the stroma. So enzymes converting 3-carbon sugar into other biological molecules are present in the stroma as well. So that's a useful thing. To have the enzymes that are going to make use of the photosynthetic product in the stroma is an adaptation as well. 3, let's not forget that we have these gigantic starch grains, okay, in the stroma as well. So you likely have enzymes that are polymerizing the produced glucose into starch, and therefore you get the starch grain in the stroma. Now, why is that good? Because remember, glucose is a soluble molecule. So it might be great for you to be making lots of glucose in the chloroplast, but then if you got too much glucose in the chloroplast, water is going to be drawn in by osmosis and likely going to cause this chloroplast structure to burst. Okay, so what you need is you need to quickly convert that glucose into something insoluble. And when you do that, you convert the glucose, you polymerize it, form those glycosidic bonds, produce amylose, amylopectin. You have yourself a starch granule, but all that glucose is now stored in an insoluble form, which does not cause water to come into the chloroplast, or doesn't affect the water potential of this structure, and therefore it's a way to store lots of glucose without causing this structure to burst. Okay, so that's probably an important reason why that's there. Okay, now remember, as glucose is needed, it's not just the glucose that's utilized here. The glucose produced by the hydrolysis of the starch is going to be exported into the cytoplasm so it can take part in glycolysis and then respiration. So for that purpose, you have likely got transport proteins in the outer and inner membranes to allow that to happen. Okay, so you've got the starch as glucose storage. Okay, because we appreciate the importance of not storing loads of glucose in the stroma. All right, next, 4. We're getting to the end now. 4 is lipid droplets. Now, in my research, it seems a little bit that the presence of these lipid droplets is a little bit controversial. So some people say that they do exist in the chloroplast, and other people say that maybe that's artifactual. But here's one reason why it's probably a good idea for these lipid droplets to be there. That fatty acids might not be immediately transported out of this chloroplast. In the event that you've got lots of fatty acid production, the thing that's going to happen is, remember that the fatty acids are acidic. Okay, they have a carboxylic group. So fatty acids, remember, long hydrocarbon chain, but at one end you've got the acid group, carboxylic acid group. Now, you probably don't want lots of accumulation of that, because that's going to affect the pH of the stroma, and that pH will affect the functioning of the proteins in the chloroplast, and so we don't want an accumulation of fatty acids. Therefore, it's probably a good idea to convert those fatty acids into, or attach them to glycerol and convert them into triglycerides or some other form of esterified lipid, okay, so that you don't have this effect on the pH, which could potentially affect the functioning of the other proteins. So you probably want to have a way to convert fatty acids as they're produced into a kind of inert, less reactive or less influential molecule, something like lipids, all right, which don't affect the pH of the chloroplast and allow the chloroplast function to proceed. So ladies and gents, we got there in the end. Thank you for persisting all the way up to this point. I hope you found it useful. I really do believe that it's not really worth saying anything unless it takes a long time to say. So if you do have any questions on this, please put it in the comment section. When you ask questions, that's when you're really making some progress. Okay, fixing things as you go along, identifying the things that don't make sense, finding reasons for the way things are, that's when you're really learning this stuff. So I hope you have found this useful. In the next episode, we are going to be looking at the cytoskeletal elements of the cell, actin filaments, microtubules and intermediate filaments, and related structures, so cilia, flagella, centrioles, things like that. That will all be covered, and that should be the last video in the episode 4 series looking at cell structure. Okay, guys? So, thank you very much. I will sign out. Good luck.